Welcome to Inert Chemistry. In today's video lecture, we are discussing about the salvage pathway. We will see what is salvage pathway, what is the difference between salvage pathway and de novo synthesis, and importance of salvage pathway. Salvage pathway. Salvage pathway refers to the process that utilizes nucleosides and nitrogenous bases that are available through the diet or from the polynucleotide degradation, means degradation of DNA and RNA to produce purine nucleotides. It is a minor pathway. It uses free nitrogenous bases and nucleosides to prepare the nucleotides. It is important in certain tissues like brain and erythrocytes in which de novo pathway is not possible. Salvage pathway occurs in certain tissues such as bone marrow, brain and erythrocyte. This pathway used the preformed nitrogenous bases and nucleosides. Here you see that nucleotides, they undergo breakdown and they are broken down into nucleosides and nitrogenous bases. You know that a typical nucleotide is made of three things: nitrogenous base, sugar and phosphate group. When it broken down into nucleoside, the phosphate group is released from it and only nitrogenous base and sugar remains. While further breakdown occurs and the sugar molecule is released and only nitrogenous base remains. So body utilizes these nitrogenous bases and nucleosides and again resynthesize the nucleotides. For example, if body use nitrogenous base and it replenished or resynthesized these nucleotides, it uses PRPP. This PRPP is phosphoribosyl pyrophosphate. This phosphoribosyl pyrophosphate, it donates ribose 5-phosphate to a nitrogenous base and again the nucleotide is formed from it. The enzyme involved in this is phosphoribosyl transferase. Similarly, the nucleotides can also be synthesized from nucleosides. In these cases, ATP are the phosphate group donors and by the help of enzyme kinase, the ATP donates its phosphate group to nucleoside and again nucleotide is recycled or replenished. Here we see the difference between de novo pathway and salvage pathway. De novo pathway of purine nucleotide synthesis refers to the process that utilizes small molecules such as ribose sugar, carbon dioxide, amino acid, etc. to produce purine nucleotides. While in case of salvage pathway, the nucleotides are synthesized from nucleosides and nitrogenous bases. De novo pathway is a major pathway while salvage pathway is a minor pathway. In de novo synthesis, simple compounds are utilized, while in case of salvage pathway, free bases and nucleosides are utilized. Important, de novo pathway is very important since it is the major pathway of producing purine nucleotides. Salvage pathway, it occurs in certain tissues like brain and erythrocytes. All cell types have the ability to carry out de novo pathway. Salvage pathway occurs in certain tissues like bone marrow, brain and erythrocytes. This de novo pathway do not use the preformed bases and nucleosides, while salvage pathway use the preformed bases and nucleotides and nucleosides to prepare the nucleotides. Here we see the importance of salvage pathway. The salvage pathway, it recycles the nucleosides and nucleotide bases that are formed during the nucleic acid breakdown. This pathway reduces the energy and resource requirements for de novo synthesis. In de novo synthesis, the nucleotide formation occurs from small precursor molecules that require much of the energy and precursor molecules for this. But during the salvage pathway, the pre-existing nitrogenous bases and nucleosides are converted to nucleotides. That reduces the energy and requirement sources for de novo synthesis. This is all about the salvage pathway. In the next lecture, we will discuss about the salvage pathway of purines and salvage pathway of pyrimidines in detail.